on your apologize for the video.
Should I stop here?
Tell you about another tissue where the local mechanical properties are important. So this is cartilage. These are some of the absolutely stunning images that we've gotten in our lab. It's a beautiful tissue. It has a depth-dependent structure, and for that reason, we sort of got into the game and started working with people in the mechanical engineering department to try and measure those properties. So, if my presentation will go to the next slide. Okay. So we all know about cartilage. It's the tissue that coats the bones in our joints. We know actually quite a bit about the gross mechanical properties of the tissue. It's viscoelastic, and in the right geometries or the right compressions, poroelastic. We know it has a depth-dependent structure, and we know a lot about the chemical nature of healthy and diseased cartilage. Despite all of this, we know actually very little about the local mechanical properties. Okay, and we know, for example, that there is a depth dependence to the concentration and orientation of the different components that make up the cartilage. And so, for example, near the surface, you have collagen fibers that are arranged parallel to the surface. Near the bone, they're arranged perpendicular to the surface. They form the extracellular matrix. There are cells, and there are these proteoglycans, these charged sugar molecules that essentially hydrate the tissue. And we know a little bit about how cartilage absorbs shocks in compression. So the idea is that this is sort of an old, I guess, one of the oldest versions of a shock absorber, where if you try to compress the tissue, the water has to flow out through a porous network. And so then you have some time scale for that fluid to come out, and so you get a very poroelastic-driven phenomenon. So, we've been interested in these kinds of shock absorbers for a long time, ever since we've had stagecoaches and buggies. Here's an example of a slightly less old, but still, fairly old shock absorber. Here you had some mechanism that translated vertical shocks into friction. And then the modern shock absorbers actually work very similarly to cartilage. They have these little holes in one of the pistons through which the fluid flows, and that's sort of defines the time scale of the poroelasticity again. But it turns out that if you look at cartilage, the main failure mode for cartilage is shear. And so then you start thinking about, well, how do we think about shear absorbers? So this is like a really old one. This is the mausoleum of Cyrus the Great. And the idea is that this mausoleum was built on very smooth stones. It's in Iran, which is prone to earthquakes. And so when the earthquakes hit, the mausoleum actually shifts on these smooth stones. And that was the idea then. These days we'll have buildings actually sitting on top of shock absorbers, or I guess shear absorbers, where the outside is made out of rubber and the inside is actually made out of lead. And then when an earthquake hits, these things end up taking up all of the strain, and then the building on top doesn't move at all. So what we want to do in our lab is ask a little bit about how does this kind of shear absorption happen in the tissue. Okay, so we take cartilage tissue from the patellofemoral groove of a young cow. We dye it and treat it with antibacterial, with antibiotics, and essentially then put it in some salt solution. We then look at the tissue in our shear cells. So the idea is that if I have a tissue, which here's the surface, here's the bone, I take it, I put it on its side, and in my shear cell I move one of the plates back and forth, and I measure the force on the other plate. And then I look below at the tissue, and the idea is that any parts of the tissue that are softer than another are going to end up being are going to end up straining further. Okay, so parts of the tissue that are stiffer are going to have a much much lower strain associated with them. So here's the kind of thing that we see. So this is a video. Let me show you what it is that you're looking at. These dyed parts are the cells. This part is the surface of the tissue. About 5 pictures like this below would be the bone. All right? And so the idea is that if you take a look at the movie, this is a looped movie, At the very beginning of the movie, what you see is that the cells near the top move faster than the cells below. But towards the end of the movie, everything starts to move together. Okay, so I'm going to repeat that a couple of times. Yeah.
What's the dye?
The dye is something that they had on the previous page. I don't know what it is. Let's see there. Blah, blah, blah. It's this stuff. Okay? It's some dye. It basically, it goes into the cells and then it can't come out. It reacts with something in the cells and it can't come out. Okay,
So what's between the cells?
So between the cells is an extracellular matrix of collagen fibers and these proteoglycans. So these are the charged sugar molecules. Okay? So they draw in water because of their charge. And so again, so the idea here is that if you look at the beginning, these cells move fast, and then towards the end, everything moves together. Okay, so in the beginning, these cells move fast, and then towards the end, everything moves together. And now the beginning, these cells move fast, and everything moves together. Okay, so the idea here is that automatically from a video like this, you can already tell that the tissue is not homogeneous, that right over here, there's a region which corresponds to, it turns out, where the collagen fibers are randomly oriented, which is much, much softer than the tissue above or below at very small strains. But then as you strain the tissue further, that region becomes stiffer, and then everything starts to move together. Right? So that's what you're seeing in a video like this. And you can quantify that, so you can, for example, use PIV to track the displacement profile as a function of depth, okay, in the horizontal axis. Then from something like this, you can take the local derivative and get the local shear modulus, or the local strain rather, and from the local strain you can then extract the local shear modulus. And so this is just telling you what you see, what you saw in the video, that near the surface the tissue is quite stiff. It took this massive dip right below the surface, about 100 microns below the surface, and then the tissue became stiff again as you went down further towards the bone, okay, in the bulk of the tissue.
These cells are dead, right?
No, the tissue is alive. Turns out chondrocytes are not vascularized, so they can survive for a very, very long time. They're like cockroaches. They just don't die. So the way, and so then you can quantify further what I showed you in the movie, which is that if you take a look at that dip, as I go to higher and higher imposed strains, that modulus creeps up, and eventually that dip disappears, and you just have a region that's as stiff as the rest of the tissue. Okay, so that's what this part shows, and I think we have the statistics here. So if I plot the minimum modulus divided by the deep or plateau modulus, you can see that you get this very statistically significant difference between them for small strains. You can also show another experiment, which is that if you take the tissue and you first compress it, then this weak region actually gets deeper. So in other words, the weak region gets weaker. And so when you start to put all this stuff together, you come up with these thought models, and the thought model that we came up with was something like this, where the idea is that if you have fibers that are connecting the top of the tissue, they're parallel near the top, perpendicular near the bottom, and if I now start to compress the tissue, these fibers might buckle. And the idea is then that buckled fibers are very easy to strain, because if I have, I'm going to borrow Jack's power thing here. So if I have some kind of fiber here, it's very easy to shear it when it's bent, but the minute you strain it enough so that they become taut, then everything starts to move together. Okay, so that's the idea behind the network here. Of course, our network is much more complicated, and we're going to need a lot of help from the folks who do these semi-flexible polymer physics type problems to try and figure out how this extracellular matrix of collagen fibers, which probably looks much more like this, with changing orientations and changing concentrations, provides you that kind of response in the macroscopic bulk tissue. Okay? So we did one other set of experiments that I want to tell you about. We looked at the frequency dependence, and now we only applied strains that were small enough so that we were in the linear regime. So we didn't go to high enough strains where the polymers were stretched. We just went locally and asked what's the frequency dependence of this tissue. And then we plotted that as a function of frequency for the minimum region, that weak region that I showed you, and the bulk region, the stiff part. And what we saw was that the stiff part was, of course, much higher in value than the weak region, but it looked pretty much like a plateau, and then a very small region over which you got a non-trivial frequency response, and then a plateau again. And so what this kind of curve tells you is that there's only a limited range of frequencies over which this region of the tissue is actually viscous. Okay, is absorbing, has a non-trivial loss modulus. Whereas if you take a look at the G-min, which is associated with that dip at 100 microns below the surface, it has a non-trivial frequency dependence all the way through, which tells you that the G double prime, the loss modulus for this part of the tissue, is again a substantial fraction of the modulus. Okay, so why is that interesting? Well, if you start to then ask about energy dissipation in the tissue, what that's telling you is that since energy dissipation goes like the loss modulus times the strain amplitude squared, this weak region ends up being where almost all of the energy is absorbed in the tissue. Okay? So yes, we know that cartilage absorbs energy and shocks when that are applied, between bones, but where in the cartilage is this energy absorbed? And the answer is in this weak region right below the surface. So why is that important? Well, because there's a lot of procedures these days that you can go into, that the doctors will do, to shave your cartilage and do some resurfacing. And if you start shaving too much, i.e. more than one hair's diameter, then you've pretty much gotten rid of this weak region, and you've set yourself up for later trauma, to your cartilage. So I think what this work is trying to get at is that there's quite a bit that we can learn from trying to look at the local mechanical properties, and we're going to need to understand the local mechanical properties in order to address how healthy tissue functions, and then say how diseased tissue functions differently, in order to then eventually come up with artificial tissues that we can construct in order to address these problems. I just want to say that once we started doing the articular cartilage, which is the cartilage of the knee, people started coming to us with all sorts of other cartilage. So this, for example, is cartilage of the spine. It's the intervertebral disc cartilage. Now, I'll just show a couple of movies before I end, but the idea here is that you have a tissue which is composed of lamella. These are fibers that are oriented in pleats that are surrounding some nucleus. And the idea here is that when you shear this, in this particular configuration, you're shearing this way. And so if you can look at the little wavy lines in there, as I shear one direction, they straighten out. But the other side compresses. When I shear the other direction, this side straightens out and this one buckles. So there's this kind of double rope-like, mechanism. Why is this the way it happens? I have no idea. What about if you then take a look at a different orientation, where now instead of looking at it like this, I'm putting it down so that you're looking at fibers coming out of the board and fibers that lie along the board. So here are the ones that lie along the board, here are the ones that are coming out. And what we find is that in this configuration, only the fibers that are coming out of the board, my little fingers here, are absorbing any of the strain. Again, a very non-homogeneous response. Why is this the best way of constructing a tissue to allow us to do, the various motions that we know how to do in order to attract the other sex and then reproduce and whatever? So again, the idea is that studying the local mechanical properties ends up being very important for addressing these kinds of questions. So I think with that, I'll end and just say that we're, I think, really just at the very tip of the iceberg, because pretty much all the tissues in our body are inhomogeneous locally, and so trying to understand something about them would be useful. Thanks.
Have you put these things under tension? Have you put these under tension and then?
Yeah, so you can put them under tension if you have glue. Otherwise, we're always putting them under a little bit of compression. And with glue, you can put a little bit of tension.
And then does your thought model sort of...
Yeah, so what we did was we showed that if you... so we didn't do it under tension, we did it under compression. But the idea is that if you compress the tissue, then you buckle the fibers, and then you have to go farther in strain before you get the strain stiffening. And that part we get. Okay, the opposite, which is what you're suggesting, which is to pull it, you also should go a much shorter distance. Okay, so we do see some of that, but we've only gone through the compression part. We haven't done the tension. There it is.